All right, now it's time to move on. I see. The first of our speakers is Chris McFarland, talking about quantifying the cell-intrinsic, cell-extrinsic growth effects of cancer-causing mutations in vivo.
There might be a title slide, I think, just one. Cool. Oh, I guess I have control over it too. Cool. Thank you for that great talk and that introduction. So we're the Cancer Evolution Group at Case Western. We're a brand new lab, or 2 years old. And I'm going to tell you about some work that began as part of a K99 project that we're trying to kind of wrap up here. And it's been a really great treat to come chat with you all. It's been a fun conference. All right, so most of you are familiar with studying cancer as an evolutionary process. Some of the people I talk to are less familiar with this. The reason why we're able to apply evolutionary theories to cancers because cancers have all the ingredients necessary for evolution. Tumor cells mutate epigenetically and at the genetic level in this very complicated tumor microenvironment that we've been chatting about, and I'll go into in greater detail. And that environment selects for certain phenotypes or traits over time. And this process of natural selection is adaptation through natural selection is really what mediates evolutionary theory. So during my postdoc, I joined the lab of Monty Winslow and Dmitry Petrov, and our goal was really to try to make mouse models of tumorigenesis more quantitative and genomically comprehensive. And so Monty studied lung cancer, and one of the main ways people would study tumor initiation using genetically engineered mice was to deliver lentiviral capsids to the mouse's lung, which then activated oncogenic mutations in the bred into the mouse's germline. For example, this is an oncogenic form of KRAS. When Cree activates this oncogene in this mouse epithelial cell, it causes increased proliferation. And then if you also activate a fluorophore, a red fluorophore, people were able to sort of visualize this widespread hyperplasia across the mouse's lung. And this is really how people studied growth in mice when I began my postdoc. So if you zoom in, you can see there's these very sporadic little dots. And the way people quantified it was simply by just sort of circling them and trying to see, what happens. So you can see if we knock out the most common tumor suppressor mutation in lung cancer, we get bigger circles, but there's a tremendous amount of growth heterogeneity. And we really wanted to try to understand these mouse models more quantitatively. And we did that by making one simple change. We added random DNA barcodes, 20 nucleotide long, random sequences of A, T, Cs, and Gs to each viral capsid. And then when the viruses stably integrate into the mouse epithelial cells, the barcodes get carried along during cell division, such that at the time of euthanization of the mice, we have different tumors, each uniquely labeled with their own DNA barcode. And we can very precisely quantify tumor size by simply extracting genomic DNA from these mice, amplifying the barcode regions, and deep sequencing, and then deep sequencing. And we can spike in kind of a ladder to infer an absolute cell number from barcode reads. And this work was done in conjunction with two great grad students in the Winslow lab. So that mouse that I showed you earlier, this is what the spectrum of tumor growth sizes are using DNA barcodes. And we call this technology TubaSeq for tumor barcoded sequencing. You can see from that there is this tremendous variation in tumor sizes after just 3 months of growth. Really, we can even go down further than this, and we see 10,000-fold variation in sizes. If I then look at that next, and we also kind of looked at histology. Even these very small tumors, which are less than 1,000 cells, due to the eyes of a pathologist look like they're the precursors of carcinoma. If I wait 2 years with this mouse model, we will see invasive disease. And if I then look at that second mouse model that I discussed, where we knock out P53 and activate KRAS, you can see that sort of progresses tumors further along. And the main change in the spectrum we see is that there's this increase in rare but very large tumors when we knock out P53. If we then knock out the second most commonly mutated tumor suppressor in human lung cancer, LKB1, we see increased growth again, but it's a very different shape to the growth dynamics. So instead of having these rare but large tumors, we see kind of a wholesale shift in the mean size of tumors. And there's an increased abundance of intermediate sized tumors. So one of the most natural questions to ask after looking at this kind of data is, how did the tumors get here? What is their growth trajectory that kind of causes different tumor genotypes to really take off on rare occasion versus just a mean shift in growth? And to understand that, we just decided to think about the two most simplest models we have for tumor growth, the really growth of any evolving population, and that's exponential growth and logistic growth. Exponential growth is really just the ideal gas law for population dynamics, where cells just kind of divide and die independent of one another. And then in logistic growth, that's really the volume of the container and how I sort of think about population dynamics. So there's this carrying capacity, where as a population starts to grow further and further, it eventually fills the space or exhausts the resources or signaling molecules or whatever the case may be, the limit size. And so I want to just ask the audience now, based on that earlier data, how many people think tumors grow exponentially? People, anyone? Okay, we got one vote there. Two, okay, we got two. All right, how many people think that the tumor dynamics that I showed earlier are going to show logistic growth? All right, we got, yeah, that's pretty much the whole audience. So the answer is yeah. We see exponential and logistic growth depending on the model. So a trick question, yeah. This was kind of surprising to me, and it was kind of cool to see these dynamics. So we'll start with the simpler genetic model. This is just a single oncogenic KRAS mutation. A lot of the analysis was done by a postdoc that Jacob Scott and I share at Case, that is an independent fellow, has been working with me and Jake, Dr. Jeff Malthus. And here I'm just showing the entire spectrum plotted at 2, 4, 8, 18, 24, and 32 weeks. And to show how the spread of the distribution propagates, we'd like to look at percentiles. So I can show you the size of the 99.9th percentile, so the largest tumor out of 1,000 on average. That's that top line. You can show you the largest tumor out of 100, and everything in between. This black line is what the median tumor sizes is doing. And you can see a couple interesting features about this, this graph. So the first thing we noticed is that early on, you see this spectrum of straight lines on log plot. A straight line is exponential growth. And so we're really working to quantify a whole distribution. So there's a lot of different growth rates that we see in this multiplexed assay, a whole distribution of different growth rates that we see for this initiating tumor type. And then the second feature we see is that different tumors, that sort of at later time points, we start to see tumors start to saturate and approach carrying capacity. But different tumors, presumably depending on where that hyperplasia may be started in the lung or other features of the microenvironment or properties of the cell of origin, different tumors seem to saturate still at different carrying capacities. So we see this broad spectrum of carrying capacities sort of at later time points. We also see some of the smallest tumors start to decline over time, which was something I was very interested in as a grad student before this work. And then the next question we asked is, okay, so how did P53 create those rare large tumors? Did it increase R or increase K, the carrying capacity? Surprisingly, what we saw, I did not expect this. Part of the reason why I asked the question was just that the more complicated tumor genotype actually has a simpler growth dynamics. Really across every spectrum of the size distribution, we see pretty much exponential growth. And this was not the model I wrote up in my K99. I thought what we'd see, some of the evolutionary modeling that I'd done and others is this somatic evolutionary process where as variation in growth rates accrue over time, especially in like a genomically unstable population like P53, we'd see a fan out in growth rates. We didn't see that. Even though also in previous work, we saw that when you sequence the largest tumors in this mouse model, you do see additional driver events, loss of heterozygosity at KRAS and loss of other common tumor suppressors. And we didn't see logistic growth either. We just saw a simpler growth law for a more complex genotype. And so our sort of interpretation of this model is we saw a subtle change in the growth rates, but really this P53 just kind of blew the lid off of carrying capacity. So that it really just, we can't even really assay carrying capacity on the time scale of this experiment. And if we wait too much longer in these P53 deficient mice, they eventually die. So you can sometimes think of malignancy as growth without bound. All right, so the next thing we did was we also wanted to multiplex. During my postdoc, that was right when CRISPR gene editing had sort of come about. And so we wanted to look at the growth effects of lots of different tumor suppressors. So we created guide RNAs targeting the 12 most common tumor suppressors in lung cancer, multiplexed them together into the same viral pool, and added barcodes to uniquely identify both the lineage of each tumor growth and then the genotype. And if we look at the size distribution across a single mouse, we get these really beautiful and rich readouts of tumor growth. So on the black bars kind of show the mean effect, and then the little dots and their sizes show cell number. You can see just on the left that I've recapitulated that first experiment that I showed you. LKB1 has an increase in mean growth, but smaller tumor sizes than P53, which has about the largest tumors in the spectrum or in the whole mouse. But every tumor suppressor in this survey really has its own sort of story about growth. And so it's a really rich technology, I think, to study tumor growth dynamics. We've been using this technology to look at pharmacogenomic mapping, or two talented postdocs in Monty's group, Ann Wei Lin and Chuan Li, have done this, where we kind of look at commonly prescribed pharmacological treatment or therapies, and then look at how different genetic mutations mediate these effects. We really weren't expecting to see so much heterogeneity when we first did the study. If you were to look through the literature for known genetic interactions with the CDK4/6 inhibitor, palbociclib, you would have only identified two genes, CDKN2A, which is upstream of the inhibited driver gene, and then RB, which is downstream. And they have exactly the effect you would expect. The upstream gene is sensitive, and the downstream gene, RB, is resistant to the targeted chemotherapy or molecular agent. But really, it's just the tip of the iceberg in terms of the size, in terms of the pharmacogenomic interactions that we're seeing. And this has really gotten a lot of clinical interest from a number of collaborators of ours. But just going back to that size profile, I can look at one of the fastest R-promoting genes in that survey of 12 genes. This is loss of CDKN2A. And at just 4 weeks of time, we see across replicate mice, we see this reproducible shift in size distributions time and time again. This is 3 mice. We did this across 6 mice, and you can see every time we can, in just a single mouse, quantify growth effects after only 4 weeks. I'm just going to summarize our survey across the growth effects across the 12 genes I studied. And we see kind of 2 or 3 interesting classes of tumor suppressor genes, at least in this survey. So that CDKN2, or sorry, so we see some genes that seem to increase the instantaneous growth rate, but don't really saturate at a different carrying capacity. We call these R-only tumor suppressors. And you can see we also do not see any tumor suppressors that increase K alone, which was kind of interesting to us. We don't know if this is something that would persist if we studied more genes or not. But we see this other class of tumor suppressors that seem to increase both the instantaneous growth rate and the carrying capacity. So So it's very interesting to me to see all these different growth effects. One thing that also kind of jumps out to me in this effort is that the instantaneous growth rates are very subtle things. They're really only an increase in growth of around 5%, whereas tumor suppressors that increase the carrying capacity really do seem to have some sort of transformative effect. So we kind of are showing that there's about a 20-fold increase in carrying capacity from our statistical inferencing. But as you saw in that P53 time series, we really, these values for K are probably kind of unbounded in our analyses. So that's the conclusions from, so in this study, what we really find is that lung tumors initially grow logistically with a single mutating event that has a lot of variation in growth rate and carrying capacity, despite being isogenic tumors in the same mouse. Driver mutations can increase the growth rate or carrying capacity or both. Our technology is precise enough to measure growth rates after just 4 weeks. And we're really trying to come up with a good statistical toolkit to kind of summarize the change in the entire spectrum of growth rates and carrying capacities for any tumor genotype, because I think that's going to be a powerful readout for any sort of pharmacogenomic mapping. So I spent a lot of time today talking about this simple logistic growth equation, but as a number of speakers have talked about before me, this is really just looking at a single genotype in isolation. When you start to look at genotypes, how they interact, it's not just the instantaneous growth rate of one species or entity, but growth is a function of the stuff in your environment. We've talked a lot about it in the context of species, where the population of wolves might mediate the population of moose. Earlier yesterday, we talked a lot about interactions between different cell types, like tumor and stroma. But hopefully today I convinced you that these individual mutations within the tumor have different effects on their environment. And we really want to just try to model this moving forward. I'm approaching the end of the talk, just by doing a series of transductions where we can start to look at interactions between different tumor genotypes within the same tumor. And we'll also overlay this with spatial DNA barcode sequencing. That effort is being led by Dr. Zucker Faber. This mouse model, I think, is really powerful for programmable genetics and unknown fitness effects. We're also doing a lot of work on in silico modeling, where we can program everything about the environment, both the growth effects and the genetic history. Okay. Just want to thank the people.
Hi, what happens during those 4 weeks?
Say that again?
What happens during those 4 weeks?
So presumably there's not much time for evolution there, but this is kind of the earliest time point we've really looked at in these mice. So I think of it as just kind of independent growth. Thank you very much. That's really great. The topic to be determined between now and the time. Yes.
Hello, yes, I'm sure you've all been on the edge of your seats waiting for my title. Yeah, so what happened is a speaker wasn't able to come. So we thought that this would be a great time to insert this topic into the exciting discussion that's already going on at the conference. So disclosure, this is not actually my work. This is actually the work of Kamala Bond. She was our phenomenal MD PhD student we had that graduated. She's now doing her neurosurgery residency at UPenn. Definitely look for her doing great things in the future. But yeah, basically I'm the spokesperson today. So I am early on in the proposed author list, so I should know what I'm talking about. Is this the clicker? All right. So we're in Kristen Swanson's lab, and as many of you probably know, we work with glioblastoma. And glioblastoma is a primary brain tumor known for wide inter and intra-patient heterogeneity. It's very aggressive. But clinically, what's going on, like in the clinic? So the clinician, because of that very eloquent location of this tumor, they're not able to really have a lot of access to tissue. So pretty much through the entire course of the disease, they're really working with imaging. And this is basically what they're seeing. So on the left is a T1 GAD image. On the right is a T2 flare. The T1 GAD is basically showing them where the blood-brain barrier has broken down. And the T2 flare is basically showing them where there's extra fluid. And this fluid could just be fluid. It could be immune cells. It could be cancer cells, but they're not really sure. So how do they make decisions in the clinic? Well, what usually happens is when they go to surgery, there is a single biopsy taken from this region. And from this biopsy, they are going to make their diagnosis. So that involves like immunohistochemistry assessments. They'll probably do a little bit of genetics. They might do some RNA-seq, but basically they're going to take this one biopsy and have to make extrapolations for the entire tumor. So they have to think of it almost as homogeneous when they're making their decisions. Now, there are a few cases like, so when they're thinking about cell density, they can make some educated guesses. So they think about like the T1 GAD is probably higher cell density, the T2 maybe lower cell density, and that can help them in thinking about surgery targets and radiation targets, but they're still educated guessing and they're not really sure. And this is only on cell density. Of course, the truth is going to be much more complicated. And here I'm only showing 2 colors, but as all of your presentations, is probably more like 4 to 10 or who knows how many colors, right? So in the clinic, I'm specifically going to be talking about how this influences how they're dealing with immunotherapy. So immunotherapy is just a whole other beast because imaging is already complicated and immunotherapy takes it just to the next level, right? So here is an image of time sequence of a patient on immunotherapy. So the number there is indicating the month relative to the therapy. So -1 means we're before surgery, then 047, we're moving on. You can see on this T1 GAD image that that abnormality is getting bigger. But somehow at month 7, 9, 11, it looks like it's stabling out and then it starts shrinking. So like I said, when we're looking at these images, we don't actually know what we're looking at. What we can infer from this is that it was growth that we're probably visualizing actually an immune response that is looking like tumor recurrence, but then it's ultimately resolving itself. Now, the issue is that glioblastoma patients do not generally live very long. The median survival is only 15 months. And the clinical recommendation is to make your IRANO assessments or resist or whatever at 6 months with immunotherapy. So you're looking at this chart here at 6 months, what's your call going to be? It's very unclear, right? So imaging is confusing, it's non-specific, and generally it just leaves more questions than answers. So is there something that we can do to make this a little bit better? And what will that take? That's what I'm going to be talking about, one of our efforts. So I think I went too far, right? So we have been thinking like, how do you get at this? You kind of need to have more samples, right? You need to know more about what you're looking at. And over the last 5 years, I'm not going to spend too much time on this because there's a whole talk tomorrow on it, but we have been collecting a really fantastic set of data of image localized biopsies. So what this means is that the surgeon is taking tissue from the patient, and we actually know approximately where on the image is coming from. And this is really exciting. We're getting more than 1 per patient. So on average, we're actually getting about 4. And yeah, so we're seeing what we can do with that. Of course, our big vision, the big dream is that basically through this, we're going to be able to connect the biology to what we're seeing on the imaging, right? So the idea is you have your biopsies, then we have all this different clinical imaging that's available for these patients on our trial. You're able to figure out like, whatever biology you're interested in, histology, genomics, transcriptomics, and then connect it to the regional imaging features through the power of modeling, machine learning. We're hoping then that we can train a model to give you the regional imaging, the regional information about the underlying biology from those images. So that's our goal. Now, in this talk, since we're looking at immunotherapy, we're specifically going to be hoping to model where T cells are. So we're going to be looking at transcriptomics. Now, the thing is with these biopsies, they're, when you do any sort of analysis with the tissue, it's destructive. So you can really only do one. If you can cut it in half, you can do a couple things. But so what we generally do with them is we do RNA sequencing, bulk RNA sequencing. But I want to know about T cells. How do we get down to just the subpopulation and really thinking about that? Well, we have been using something called CyberSort. Another version is CyberSort X to deconvolve these cell populations. And just real quickly, the general idea is that this whole algorithm is trained ahead of time with, you have some single cell data, single cell RNA sequencing, and you get some idea. Each of the cells have been tagged as a type of cell that you think is going to be in your sample. And then you get an idea of the transcriptome profile for that cell type. You can then create a gene expression signature matrix that you think, ideally, if you were to multiply that by a vector, that's the percentage of those cell types in your sample, you would ultimately get to realize your bulk RNA sample. So basically, this is looking at an AX equals B kind of situation. So it's an inverse problem. It is overdetermined, very much so, but you can do some regression and you can get an estimate of the cell populations that are in that sample. So that is what we've been doing. The library that we were using for the single cell was a default library in the CyberSort. It was leukocyte, so I think is pretty focused on the hematopoietic system, but it breaks it down into 22 cell populations. 7 of them are different T cell subpopulations. So when I talk about T cells later, I am going to be talking about the sum of all of those T cells. All right, so just recap. What am I trying to do? Because there's a lot going on here. All right, we have MRI localized biopsies. That means we have a biopsy. We know where it is in the brain. So from the imaging part up on the top, we think about where it is at each of those spots for the biopsy. We are going to be calculating, figuring out a lot of the features there. So feature extraction, there will be feature selection there. On the bottom, we also have tissue, and we are doing RNA sequencing and then deconvolving it into the different populations. And then we are doing a radiomics model, in this case, an SVM regression for predicting the T cell population at each voxel or at each biopsy. So how did we do? I'm going to go ahead and say we did pretty good. So this data set, we had 147 biopsies. You might think that's a lot. You might think that's a little for machine learning, but that's what we had. 81 were male, 66 were female. As you just heard, we have a great collaboration with Josh Rubin, and we are very focused on sex differences. And we do think that there is reason that the imaging and, well, the biology would likely be different, and thus the imaging to biology would be different. So we did train male and female models separately. Anyway, the top is our training data. And as you can see, the x-axis is the predicted, the y is the actual, and we're doing pretty good job hitting that identity line. So we're really excited about that. The test data is still also doing pretty well. So I would call this a model that has hope, right? But any model could have hope at this stage. But the real question is it functionally useful? When you take it to the clinic, does it mean anything, right? So we were fortunate enough to be working with a clinician at Mayo Clinic who had this phase 1 dendritic cell vaccine trial where there were 20 patients on it, and the trial came back as negative. So that means on average in this 20 patient set, there was no improvement in overall survival. But, you know, with every single clinical trial, there is the question, was there a patient we missed that actually got benefit? Is there a better way to think about who should have been on the trial in the first place? And so we were thinking, if we applied our model to this, is there something like that we could find and figure out? So that's what we did. So we took each of those patients. We had a pre-treatment image and we had a post-treatment image. We segmented out the tumor regions on both of those. We then applied our model on this, and then we could make our prediction for each voxel, how many T cells we think were there. And for this initial pass, I will say that we weren't doing very spatial, like voxel to voxel comparison. We instead were doing like a sum of all the T cells pre-treatment, a sum of all the T cells post-treatment, and we were looking at the variation between that. So for this specific one, we were also focused on T1 GAD and T2 flare images only. That first slide before had a few other images, but these are the two that are available pretty standardly clinically. And that is also really important for these kind of models. So what we were wondering is if we look at it, like if it goes up or down in the T1 GAD or up or down in the T2, what do we see? And what we found was that if you think about that, like that kind of quadrant over there, that if you are in the situation where your T cell population was predicted to go down or be stable in the T1 GAD, but go up in the T2 or flare, then we were actually seeing a differentiation in the survival of the patients of those two cohorts, if you were in that quadrant or if you were not. And in fact, in a Cox proportional hazard regression analysis, that was the only thing that came up as significant in this survival analysis. So I would say that this is very exciting, tantalizing data, but of course there's always more work. Yeah, so in summary, interpreting clinical imaging is challenging. Just period. We have trained a model to predict T cell abundance based on the clinically available imaging, and it does seem to have some discriminatory ability. But the future work, always more data, right? So we trained this on 147 biopsies. We actually did recently get another 50-ish analyzed with bulk on RNA-seq. We're hoping to get even more than that. Hoping to get more funding for some of that. And we also have connections with a few more immunotherapy trials and their imaging protocols, so that we're hopefully going to apply it there and see if it is more broadly deployable and interesting. So with that, thank you, of course, to all of our funding for this. Lots of NCI support, lots of James McDonald support also. And then this is our team. And again, just really want to point out, this is Cam Bond's work. She is amazing. She does great things. And thank you. Any questions?
Yeah, that indeed is amazing. Have you considered using imaging-based flow cytometry to identify T cell presence rather than the RNA-seq approach? Because that would be much faster, and I think the technology is available now. So
It would be much faster?
So you can do this in real time, right?
Oh.
So you take the biopsy, and within 15 minutes, you know exactly how many T cells you had where.
So
Yeah.
tomorrow during the talk where they're talking about when we collect all the data,
Yeah.
you'll get to hear about all of the challenges of in the OR room, what's going on.
Yeah, awesome.
So, I mean, yes, I would say that we're always open to discussions of how to make that faster, quicker, better, faster. But there are logistical challenges in the OR room.
I can only imagine.
Yeah,
but
yeah.
super cool.
Okay. Thank you.
Talking about calibrating tumor growth and mutation parameters, the spectral spatial analysis cancer biopsy.
Thank you very much. My name is Stefano Posetto. I work in as a scientist at Moffitt Cancer Center, and I'm going to show you some results of analysis immunohistochemistry slides. First of all, some, let's say, motivational slide. So in 1975, before most of us were born, 1 in 11 women were diagnosed with breast cancer. And nowadays, despite a growing rate of 5 years rate survival of 95% plus, still 1 in 8 women and 1 in 1,000 men are diagnosed with breast cancer. So it's, I know that I'm probably the last obstacle from your path to the outside lunch, but just to translate this number in, these are only US figure, but if you think at general worldwide numbers, by the end of my talk, 65 people will be diagnosed by breast cancer. By the end of my talk, 19 people will be dead because of breast cancer. So try to think that we are here for not just show off how cool we are, but to remember that we are trying our best to solve this problem. And so motivational slide, surely, oh, wait. From my side, the investigation, how this, the nature of this disease changed the structural and the morphological evolutions of the breast. Here in this slide, you have a normal breast architecture after on the top right, with the presence of carcinoma in situ, when become invasive, you see the bottom right, until when become metastatic. So clearly looking at these images, my idea was trying to understand how can we characterize the morphological evolutions of this cellular architecture, how to relate it with the disease degree, and how these theoretical informations that we can use to connect the anatomy of this pathology with this physiology. And also because I work in a Moffitt Cancer Center is actually one of the top 10 cancer hospitals in the United States. Of course, I want to try to see how my theoretical knowledge can be integrated in the decision-making process today, now, tomorrow, not in 10 years, okay? So I've been provided with this database. So if I just to study early stage breast cancer, source of adaptive radiations, 9.5 grade, three fractions, staying with the immunohistofluorescence. So basically inform my on position and locations of immunocells, most important elements, B cell, natural killers, B cell, and all the markers, CD3, CD4, and FOXP3, etc. I've been informed, given me the information for pre and post treatment with these slabs that as an example of a section area where I can identify a region of interest as a tumor interface region between tumor, stroma, and stroma areas. Okay, so that's what they look like. There's not the table here. I don't know why it doesn't appear. So, so I apologize, there's no table. There's a piece of table there. So anyway, I try to focus myself only between these set of patients, only with the patients for which I have both pre and post. And I will show you the results on 6 of them. To start, what I decide to use, which tools can we use to code this distribution of cells in an information that we can read? How can I get? Can I extract information from this cell distribution when you give to me? Well, one of the tools that I loved for this project was the 2-point correlation function that basically tells you how many stars are around another one, okay? So if we are completely distributed randomly in this room, we maximize the entropy, there's the 2-point correlation functions is basically a line around 0. If we are more clustered than random, we are above it. Under clustering, we are below 1. It's not really a correlation, as you see mathematically, is between -1 to implies infinity, so it's not upper bounded. In particular, a useful tool, nevertheless, that I'm going to show you today, works with its Fourier transform. It's called power spectrum. It's basically inform you on how much energy the cells are using and having in their normal cycle per unit of space, okay? What you see in the bottom is a wave number, something proportional to the frequency, inversely proportional to the dimensions that they're using. So power spectrum is a powerful tool to understand the evolution of the cells across many different size of the cell. I'm considering between 4 and 40 micrometers, okay? How, well, yes, of course, there are also, you see the slides are not infinite. There are some limited, and for the reason that I'm going to show you, I'm interested to see what's the difference between this clustering compared to a completely random in an infinite set. So I'm using torus topology, a fancy name, nothing scary. That means that I'm basically repeating infinitely the slabs that I have, the region of interest along the directions. And how do we interpret it? Well, the energy with which these cells will evolve actually will depend on the ability of them to move, so something proportional to an evolution of time or this. And if I assume this torus topology, I will assume that basically I'm interested in the difference between a completely random evolution. So the chemical potentials that move them is proportional to with the factor, the diffusion factor, that is a scalar function for me. On their ability to grow can be accounted for with many different general law. Because I'm interested in a set of cancers, I will consider just an exponential growth. So the interesting results, this is a tough math, but somehow you have eventually worked out, and you discover these interesting results you see here below. This is a power spectrum of the reaction diffusion equation. As you see on the left, the contributions of its shape is only for small wave number that if I vary the growing factor, and is only from different diffusion coefficients if I look at the large wave numbers. So what does it mean? That I've been given a completely disentangled system between reaction and diffusion coefficients, if I am able to fit, and if it looks like the observed one. For free, I didn't know in advance. I had to show this equation. Does it look like with the same power spectrum that I find from the observation from the slab? Yes, the answer is yes. Here you see pre and post immunohistochemistry analysis of power spectrum in blue and red respectively, or vice versa. And the line that you see passing through it, it is the power spectrum from the reaction diffuser. So from a single slab, I can evolve the tumors. I don't need to from a single shot. Note that either you're right or you're wrong, because the diffusion at the way high weight numbers change can be shifted, but the slope cannot be touched. So if you are right, as you see here, I fit it, you get the correct theory. Otherwise, the theory is wrong and you see it immediately, okay? So how does it help me to inform my oncologist coming out to ask me some information? Well, you can imagine that if I have a patient and I take the resections area and I do my analysis of it, if I find that there is a high diffusion value, then I can imagine, well, you know, this cancer is very much spreading. I might want to attack it with like a systemic therapy, like a chemotherapy. Which means if I have a very strong, high growing factor, well, this cancer is in the breast is growing like a balloon, better to attack, for example, with high radiotherapy, okay? How does it take to go from these to true applicable results? Well, in a single word, it takes money. Okay, so you have, of course, to extend the database. You have to use statistics to database. This is an extended database that I'm using. As you see here in black is the cancer on different colors, the different markers, CD3, CD4, CD8, etc. And the reason is why? Well, because I can do something like that. So I can have a probability distribution functions of the distributions of, for example, the diffusion coefficients. If somebody enters from that door in the hospital and I do my analysis and I get that the diffusion coefficient is -6.5, well, then it's poorly diffusive. But if it's -5, well, that's to be considered very diffusive. So I can suggest the oncologist today that it's better if it attacks, you know, with some systemic value. The same can be done with a growing factor. Of course, it's not published, so that's discovered on purpose. What is also most interesting, what gas can to me for free? Well, of course, you are, you know, that the tumor infiltrate lymphocytes are for some type of tumors, for some kind of cancer, informative of the prognostic, let's say, of the evolution of the cancer. So in this first, the slide that I showed you before, I can ask myself, not what's the correlation between the power spectrum, what's the 2-point correlation functions, but what is the 2-point cross correlation function? So if you are here distributed, I can ask myself, how is enhance the probability if there's a man there that there are more female around? How much is increase the probability that if there are cancer around there, there is a CD3, CD4, CD8 there around? Okay, so another follow-up work that I'm going to work is exactly on the cross spectrum. So to try to understand, to quantify the reason why there is the tumor steals in this analysis. So, of course, final point is, well, I want to eventually introduce these things in a tumor, let's say, in a decision-making framework where I've been developing much long in the past. And yes, to inform our clinicians actively on what eventually they can, which therapy is probably to be most effective, probably. Okay, that's all. I thanks Dr. Mendering, Dr. Gettenbig for supporting that past 3 years and a half, and I'm also on the market. So if you're interested to support this research further, please follow me and contact me during lunch. Thank you.